lot of studies that focus on identifying the function of different proteins. A lot of the time, this is done using the similarity of the amino acid sequence of these proteins. Sometimes there are proteins that are very similar to each other in sequence, but actually control very different functions. For example, bacteria use very similar proteins to sense and respond to their environment. And while one pathway might respond to sugar and upregulate metabolism genes, another pathway might be sensing antibiotics and upregulate some set of antibiotic resistance proteins. It's very important to be able to tell the difference between these two sets of proteins because we want to make sure that we're studying the correct pathway. We might know the function of one of these pathways based on experiments in one particular bacteria, but when it comes to the other bacteria, it might be very hard to study using experiments, and so we'd rather look at it from a computational angle. However, we might not be able to tell the difference between these two pathways based on their sequence alone. And this is especially important when we are studying many bacteria that all respond to these different things. My approach is to look at the surrounding genomic information in order to tell the difference between two sets of proteins. Although they're very similar by sequence, there might be some set of conserved genes in their neighborhood that will allow us to tell them apart. I've created a program that will compare the surrounding genomic information and tell us how similar two genome neighborhoods are. Basically, we're turning the genome neighborhood similarity into a number that we can then use to build a network between the proteins that we're interested in. We can then look for the proteins with identified functions in the different clusters and use that information to give us an idea of what these pathways probably sense and respond to. In addition to identifying the probable function of these proteins, I've taken several sets of these proteins into the lab to make sure that they interact the way that we think they do, using an in vitro assay. By considering the genome neighborhood of a protein, we might be able to get a better idea of how a protein might act. Using this technique, a biologist might have a better idea about what a protein will do before they go into the lab to try and test it out. This is especially important when using model organisms because we want to be sure that we're studying the correct function of a protein when we try to apply it to human biology.